All right, go ahead and title the notes Independent Assortment. A couple points I want to make about that. First, most often, so most of the time, right? We're talking probably 95% of the time. Most often, genes will assort independently. And if you don't know what that means, you should look it up in the reading. Because they are usually on different chromosomes. So what I mean by this is that if you were to pick any two random genes, like, I don't know, name a gene, right? You could be like, hemoglobin gene. And then I'm like, okay, name another gene. You're like, I don't know, SRY gene. And you're like, okay, are those two genes, are they on the same, those two genes that you mentioned, are they on the same chromosome or are they on different chromosomes? Odds are they're probably going to be on separate chromosomes, right? Because we have 23 types. Probably the odds that they're both on the same chromosome is much less odds than that they're probably on different chromosomes. Okay. Simple concept. I just want to make sure you understand that. So if you pick any two genes at random, odds are they're on different chromosomes, which means that they can separate and they are independent of each other. But sometimes the two genes of interest are linked. Okay, so today you're going to learn about linked genes. Underline the word linked. They're linked. So what does that mean that they're linked? It means that they are on the same chromosome. Now this has some implications. I'm going to show you those implications here. It's kind of hard to visualize unless you have a picture. So we're going to draw another picture. And kind of like yesterday, you'll need two colors. Today's picture is not going to be quite as big as yesterday's, but I would pick two colors. It's going to help make your picture better. So I'll give you a second to do that. But like I said here, there is this chance, right, that the two genes that you've picked, that they happen to be on the same chromosome. It's not unheard of. In fact, like I said, maybe 9 out of 10 times they're not, but 1 out of 10 times they probably will be linked. Okay, so what we're going to do then is we're going to show both scenarios. I'd like to show you what it looks like if they're not linked. And I'd also like to contrast that with what it would look like if they are linked. So what we're going to do is we're going to draw two scenarios, analyze them side by side, and that way we can really clearly see the difference. So you got your cell, okay, and this is a parental cell, and it's gonna be producing gametes, just like we saw yesterday. We're gonna do meiosis. So say that they are in fact. Let's say that they're not linked. Okay, I'll start there. If they're not linked, then we're gonna have. Oops, hang on. That was supposed to be pink. Yeah, so if they're not linked, we will have okay, sorry for the delay. If they are not linked, then they would be located, the two genes of interest, which, by the way, the genes of interest are genes of interest. And the reason I put it that way is because usually what that means is that these are the genes that we're paying attention to. And those genes of interest is Q and R. So we're interested in gene Q and gene R. And if in fact these two genes, gene Q and gene R, if in fact they're not linked, they will be located on separate chromosomes. So that would look like this. In other words, gene Q is always located on the large chromosome, and gene R is located on a different chromosome. That's why they're not linked. And maybe the paternal set has the recessive version of each of these genes. It could be different, but we have to pick something. And I'm just going to go ahead and say that the maternal copies are dominant, the paternal are recessive. Doesn't have to be that way. It could just as well have dominant alleles for the paternal, but like I said, we got to pick something. Okay, so what would this look like, you guys, if the genes were linked? How would we draw Q and R for this scenario? Well, we would have to put both of those genes on the same chromosome. So we would do like this. That's a linked gene. The reason they're linked is because they are a package deal. They come together. They move together. They're linked. And then over here, that would look like that if the paternal copy happens to be recessive. Okay, so why do we care? Why do we care about linked versus not linked? The reason we care is because it's going to have an effect on the pattern of inheritance that we see in the offspring. In other words, it's going to be very, very likely, in fact, most of the time, we're going to find the offspring of the linked genes to get inherited together. In other words, I fully expect the gametes here to have capital Q and capital R together, because there's no way in which those can separate from each other. They're a package deal. They get inherited together. However, for if they are not linked, well, then we can get some different combinations. We could have Q and R, but we could also have Q little r. The other possibility, since they're not linked, is we could have that. And then lastly, we could possibly have this, where they're both recessive, right? So since they're not linked, we get all four possibilities because they can separate. They can get inherited either way. But in the scenario where these genes are glued together because they're on the same chromosome, we don't see that same pattern. Instead, what we're going to find is 50% of the time you're going to have both dominant, right? The other 50% of the time you're going to have both recessive. But you will never, in this particular case, it wouldn't be possible to get a dominant and a recessive because they're linked. Does everybody see the logic there? Now over here, back over here to the not linked, if you think about the percentages, it's just 25, 25, 25, 25. Okay, so what this tells us then is that if we look at the pattern of inheritance in the offspring and we see kind of a weird pattern where it looks like the alleles are getting inherited together, that's a major clue for us that these alleles are probably linked. They're probably on the same chromosome. So this is where the chi-squared, I told you there's this statistical test called the chi-squared test. And when it is particularly useful is in cases where you have an expectation and in other words, you have numbers that you're expecting. And then when you go and you get the real numbers, if they don't match, then there could be something going on. Like maybe they're linked because the numbers aren't coming out right. And so there must be some kind of linked gene thing going on. All right. So that might be confusing to you, but let me give you an example. Okay. If, so let's list out all the possible genotypes. So all of the possible genotypes is this. There's that possibility, there's this possibility, there's this, and then there's this. So there's 4 possibilities. So I'm going to go ahead and qualify this before we suggest some numbers. We're going to say 2 things. We're going to assume the genes are not linked, which is more likely than that they are linked, right? So always just assume whatever is more likely. And if I pick any 2 genes at random, it's most likely the case that they're on separate chromosomes. Maybe let's say chromosome 3 and chromosome 17. I don't know. So that's our first assumption. The second is that, well, this is not an assumption. This is just a fact. 100 offspring are produced. So the question is, what distribution of, like, how many capital Q, capital R's would you expect if the genes are not linked and they are free to rearrange? What percentage? Or what number of individuals would you expect in each of these 4 possibilities? Anybody want to share a guess? Yep. 25. Oh, yeah. By the way, this is our expected. So this column here is expected. And then over here, we're going to talk about what we actually observe. Okay. So this is, by the way, I'm teaching you the chi-squared right now. This is chi-squared. It's when you compare what you expect with what you actually see. And then we do it. We do the statistical test to see if it's different enough. So I'll show you that part in a second. But remember, Anya pointed out that if in fact these genes are not linked, we would just expect them to be just as equally likely, any combination, right? It would be like drawing alleles from a hat. And if you draw them from a hat, they're not attached to each other. The chances would just be 25% for each of those phenotypes. Okay, but what would they be if they were linked? So you look up here, right? I'll refer you back to our picture. In this particular case, if they are linked, we're going to expect 50-50 for this one, 50%, and 50% for this one, right? And then these in the middle, maybe we'll expect about 0%. Now, the reason I put the about 0 is because there actually is a way in which they could separate. And so I don't know if I want to share that quite yet. But let's just look at so let's just say that this is what we would expect. These numbers are what we would expect if they were linked. So the question is, what are the actual numbers? So let's say that the actual numbers came out to be, let's say 41, 38, and then 6 and 5, let's say. So 38, so when we actually counted our offspring, we got something like this. 38 of them were this, 6 of that, 5 of that, 41 of that. So you're noticing right away, you're like, wait a minute, it's not 50, 50, 0, 0. It's a little bit off from that. There actually are a couple individuals where the alleles did in fact separate. Okay, now the reason for that is that the reason that we have these so we're going to say some in some cases the alleles did manage to separate because of crossing over. So now you remember from the notes yesterday that there is this possibility, right, that if the chromosomes overlap with each other, they could exchange pieces of genetic material, in which case the alleles could actually swap. Okay. Now, how much is that happening here in real life? It doesn't appear to be happening all that often. Maybe there's a little bit of crossing over. There definitely is, right? Because we're seeing a small percentage of those two middle phenotypes, but it's not happening that much. Most of the time, the alleles are glued together and they're just inherited as one package deal. Okay, so the real question then here is how, if this is what we expect, 25, 25, 25, 25, the question that I have for you is, are these numbers different enough from these numbers to count as being linked? So let me just ask you, you don't know anything about chi-squared, just give me your gut feeling. Does this look linked or does it look not linked? What's your gut tell you? Like, do these numbers look quite a bit different than 25, 25, 25? Yeah, obviously, right? They look a lot different, and it looks like something fishy is going on with those alleles. Just looking at those numbers at face value, we can kind of tell that there's something going on. But what if they're a little bit less? What if it was like 32 and this was 15? At which point, what's the cutoff, right? At which point do we count it as linked or not linked? And that's where we utilize the chi-squared, is that the chi-squared will tell us the cutoff. It'll help us to decide whether or not there's a significant difference between observed and expected values. Tomorrow, you will run a chi-squared test. You're going to be doing a little experiment, flipping coins, and you're going to have an expectation, and then you're going to have observed actual numbers, and you're going to compare the actual numbers to what you expected, and then we're going to see if, in fact, there's something fishy going on with the coin flipping. So don't worry, you're going to get plenty of practice applying the chi-squared. But I wanted to show you the context. So hang on a second. We're not done. We got 8 more minutes, and then we need the 8 more minutes to finish a couple more concepts. So what this means then, the chi-squared value, it looks like this. Chi-squared is a measure of the difference between observed and expected values. Basically helps you determine the cutoff. Quote, cutoff. I don't know how else to say it, but it's like, what's the cutoff? How do we know if the numbers are different enough to count? So we use the chi-squared to figure that out. The larger the chi-squared value, the more likely they are linked, right? Therefore, we reject the null hypothesis. We'll look at null hypothesis in more detail and all of that, like all that language, right? This what's the null hypothesis? What's the alternative? What does it mean to reject it? You're going to get plenty of sample case studies and problems. But today we're just showing you the fundamentals. Okay, now I made this claim, you guys, so back up here, when we look at this 6 and 5, right? There are, in fact, some individuals in some instances where these chromosomes actually are separating. And the only way that that's possible, like if they're on the same chromosome but they separate, the only way that's possible is if there was a crossing over event. So let's look at that, okay? Draw a chromosome where Q, because we don't know how close Q and R are on the chromosome. So I want you to draw a chromosome where Q and R are very distantly located on the chromosome. They're far away from each other. So what we say is that if Q and R are far away from each other, a crossing over event is more likely to separate them. So let's show you, okay? Imagine another chromosome right here, and it overlaps right here. So what's that going to do? Well, it's going to separate Q and R. Can you kind of visualize that? Right? Chromosome overlaps. This piece down here goes over here, and so R goes with it. It crosses over, and R, that lower piece of that chromosome on the left, is going to move over and become part of the chromosome on the right. And we showed that yesterday in our drawing. Now let's compare that to the case where Q and R are very close together. It's also possible that Q and R are located close together, very near each other, in which case, if a crossing over event occurred like that, it wouldn't happen. The only way that you could split Q and R is if the crossing over happens right there, right in that little small spot. And so hopefully you can tell that that's much less likely than the above scenario. So we're going to say if Q and R are very close together, a crossing over event is not likely to split them. Why? Because there's a very narrow there's a very narrow range in which that crossing over event must happen, right? It's got to happen right between Q and R, and that's a very small target, I guess you could say. So this rate of crossing over, the rate of crossing over helps us determine determine distances between genes. More crossing over means greater distance. Less crossing over means they're closer together, right? And if you have enough information, you can actually piece together the location of all the genes on that chromosome just based on the rate of crossing over. Okay, that's going to be it for today.